So, finishing on what we were talking about last time on the adaptive immune system's antiviral response, we're going to move on to the B cells. Actually, I believe most of this is just repetitive. So all we need to know about the B cells for the adaptive immune response to viruses. They're just going to produce immunoglobulins capable of oxidizing viruses. Recall that immunoglobulin G molecules are going to be responsible for proteins, and then IgM and IgE are responsible for carbohydrates, then lipids, and then we could also have immunoglobulin E production to be integrated into mast cells. And then once they're at these mast cells, they can secrete, they can stimulate degranulation of mast cells and trigger a response at potential sites of entry for the pathogen if we're encountering it again at the mucosal membranes at the specific gates of entry. All we really need to know is that B cells can produce immunoglobulins that can oxidize viruses, and then we've already gone into depth on B cells, so if you want to know more about that, just go to the B cell videos in the playlist. So the important part of this one, we've talked about the different mechanisms that the adaptive immune system uses to deal with the viruses in the viral immune response. But viruses don't want to be dealt with. They want to be able to proliferate, infect cells, and then spread. So they come up with mechanisms of their own to try and get past these mechanisms of the adaptive immune system. And we're going to take a look at them now. So viruses are able to evade recognition by immune cells by making cell surface proteins that mimic host proteins. So they're not recognized as foreign, so they're not going to stimulate a adaptive immune response, because we selectively when we're developing T cells and B cells within the thymus and the bone marrow, and they're going through their affinity maturation process. We haven't talked about it yet, but they go through a lot of trials and exposure to antigens that make sure that none of the B cells and T cells that we produce are going to have T cell receptors or antigen binding sites on immunoglobulins that are able to recognize self-antigens, so antigens produced by our own cells. Because if they did, then our immune system would just be constantly targeting our own cell products and killing our own cells, and we would die. That is what happens with autoimmune disease when something goes wrong in the regulatory process for the maturation of these B and T cells, where we end up with some B and T cells being able to recognize self-antigens, and then that's how we get the entire problems of autoimmune diseases like lupus. So if a virus is going to create host proteins that mimic our own proteins, then it's likely that we're not going to have any B or T cells with variable regions that are able to recognize these viruses, because we select against B and T cells able to recognize these viruses, so we don't end up with an autoimmune disease. So they're kind of exploiting our own bodily processes. Viruses can also downregulate MHC expression on an infected cell surface, so that T cells are not able to recognize that the cell is infected, because they're not expressing either MHC1 or MHC2 on their cell surface. So they're not having an antigen on these cells. And because there's no antigen, that's not going to... Well, because there's no complex to begin with, you're not going to have T-cell receptor and CD8 or CD4 co-receptors forming complexes with MHC. So it basically allows virally infected cells to get past being detected by these T cells. Viruses can also change membrane proteins and molecules through kind of genetic recombination. And this basically means that because the molecules on their cell surface have changed, they're different shaped, so we need to produce different kinds of B and T cells to be able to recognize and opsonize that virus with appropriate immunoglobulin antibodies. Two important processes that we're going to go over now: antigenic drift. Antigenic drift refers to subtle change in the viral surface glycoproteins, like we just talked about, so that viruses can avoid detection by immune cells. And then they're going to need to stimulate a new adaptive immune response, because the shape of the glycoprotein surface has changed, so we need new antibodies with new antigen binding sites to recognize these new antigen shapes. And these are typically caused by point mutations or deletions, so very minor edits to the genome. Meanwhile, antigenic shift is a much less frequent process, occurring only in influenza A, and it involves profound change within the virus, resulting in new hemagglutinin or neuraminidase types being produced. And these usually occur when kind of DNA is being incorporated from two separate species. So it's a bit of a mouthful for not really much content, but this is the main thing to take away. So we went over the adaptive immune systems, viral immune responses, and the last one we talked about these mechanisms of the adaptive immune system's response to viruses. And then we talked about how viruses are able to evade these mechanisms. So that's it for that.